Okay, let's have a class. And we're going to do a very important work today for spread plating, numeration bacteria. You have the fact sheets, which is chapter 11, pore plating. We're going to do next week. And today we talk about chapter 12, which is spread plating. Okay, we're going to talk about those first. And when we finished, we're going to go over the results from 1 week ago. So that probably is lab 8, spread plating. Okay. Numeration bacteria.
Okay, we can accept that one.
Good morning.
So first of all, we start to talk about is numeration of bacteria culture. So here's a very simple question. I had a broth. We had a nutrient broth, and I had E. coli growing at 35 degrees Celsius for 24 hours. So what it looks like? It looks like turbidity. So you shake it, we talk about the 3 indications for bacterial growth. You're certainly going to see the turbidity, you're also going to see some precipitation, and some people see some of the pellicles. Now the question is, how many bacteria are there? And we mentioned about in the lecture, we said we can do like a color spectrometer. We could be using some of the chamber to do the counting. But those, we don't know live cell or dead cell. And today's methods, we can differentiate live cell and dead cell, which means how many bacteria live in this tube. So how are we going to do? Then lots of people will generate an idea to spread the liquid onto the agar plates. So what they do, they're going to add amount of the bacteria onto the agar plates. They're going to do spread plating. And then they do the incubation. These plates, they go incubation, 35 degrees Celsius, 24, 48 hours, and they hope will form in colonies. And this is what we say is CFU stands for colony forming unit. But can we do this? Not at this moment. The reason is there are too many bacteria there. How many bacteria? This is approximately 10 to the 8th to 9th cells per ml. It's huge. So if we are adding 0.1 ml, which means 100 microliter onto the agar plates, it will not form colony because it's too density. We usually call this is too numerable to count. So just simply say this is Tn, Tc. So we cannot do that. And also in the microbiology area, there is a standard number showing on the plates for the colony forming units which is acceptable. The range is 30 to 300 CFU. That's acceptable range. So how we should do? Obviously, we should do a C area dilution. So this is back to a very basic chemistry lab. How are we going to do C area dilution? C area dilution, basically there is a 2 different types. There is 10-fold or 100-fold C area dilution. So what is the tenfold serial dilution? Which means you had an original culture. You're going to prepare a cup of 9.0 ml, 0.1% nutrient broth. And how we're going to do it is we add 1 ml. Go to the first tube. Then we mix very well, we shake or vortex, and then add 1.0 ml, go to the second tube. We mix, shake very well, then we add 1.0 ml, go to the third tube. So what's the dilution factor for the first one? For the tube, will be 10 to the power of 1, 10 to the power of 2, and 10 to the power of 3. That's a tenfold serial dilution. Now, we usually also do is 100-fold serial dilution. So this is tenfold. We also do 100-fold. 100-fold. Instead of we using 9.0 ml dilution tube, we are using 9.9 ml. So how are we going to do it? We had the original tube, let's say right here. We're going to prepare a bunch of, let's say 3 9.9 ml, 0.1% nutrient broth. Then how we do? Instead of we are adding 1 ml into each tube, we are adding 0.1 ml, which means 100 microliter. So we add 0.1 ml into the 9.9 ml dilution tube, the first one, mix very well, then go here, the second tube, and do the repeated for the following, the third tube. So what's the dilution factor of the tube? It will be 10 to the power of 2, 10 to the power of 4, and 10 to the power of 6. So that's a basic idea. And finally, after dilution, we usually using these diluted tubes to do the spread plating onto the agar plates. And the agar plates, we usually add 0.1 ml, which means 100 microliter. Using spreader to do a very good job of the spread, gentle, but averagely cover the surface. And these plates, we incubate 35 degrees Celsius, 24, 48 hours. We hope they will forming these colonies. And we also hope one of the plates will fit the legal countable zone 30 to 300 CFU. Now, a couple of things I want to mention. Usually, we need first of all have an estimation how many bacteria are there. So you did the broth. Sometimes we do juice. Sometimes we do food products. We at least will have some idea how many bacteria are there. So for example, I've been working for 20 years. I know this is approximately 10 to the power of 8 cells. That's a lot. 100 million. If you're working in the juice industry, you should know approximately maybe 1,000 cells there sometimes. So that's an estimation. Second, you need to estimate how many dilution tubes you need. That's to be prepared. Third, we not only going to rely on to gambling only do one place. We usually need to do multiple dilution, and every dilution factor we need to plate it. Because we want to pick the one which is fit 30 to 300. If you're only using the highest dilution one, maybe it's only one colony there. If you're using the lowest dilution, maybe it get 400 CFU, then it's too numerable to count. So those are the two things is important before we do. And also, why we using 0.1% neutron broth instead of we are not using distilled water? Because we want to keep the osmotic balance of the bacterial cell. If we only use tap water or distilled water, bacteria could be burst sometimes. So that's why we're using 0.1% neutron broth. Okay. So this is an overview picture of what we're going to do. So today is the first time you do spread plating, and the first time in here we do cereal dilution. And I have been working for many years, so we want to make your life a little easy. So I'm going to set up the dilution sketch, and then you do it. Okay. So how are we going to do the work today? Everybody will do it individually. Everyone will get 3 dilution tubes, that is 9.9, and 3 agar plates. And a spreader, a sterilized disposable plastic spreader, and also your bacterial solution. Plus pipettes and the tips. Okay, those are the things you will get. So everybody do it individually. Now how are we going to do it? We had a broth. This broth has been grown 35 degrees Celsius, 48 hours. So I guess it is approximately 10 to the 8 cells per ml. The same as CFU per ml. So I'm going to do a dilution factor. How are we going to do it? Since it is a lot, so I'm going to make the life easy. I'll prepare 3 9.9 ml dilution tube. So every of these is 9.9 ml. I already talked about dilution factor. So you can label these as 10 to the power of 2, 10 to the power of 4, 10 to the power of 6. So how we do? You're going to add pipetting 0.1 ml, 100 microliter onto the first tube, and then mix very well. Shake 15 seconds. We don't have a vortex. So go here, we're going to add 100 microliter, shake very well, and then go here to the third tube, also 100 microliter, shake 15 seconds, do it very, very good. Now there's a tricky thing. We're gonna add into our agar plates. The agar plates is right here. Once you add into the agar plates, how many volume we use? We don't add 1 ml because it's too much. So we are adding 100 microliter. What this tells you, which means once they are added onto the agar plates, they are further diluted tenfold. So what is the final dilution factor of the agar plates? Will be 10 times more than the tube. So corresponding to the dilution factor of 10 to the power of 2 of the first tube, the final dilution factor of the plates is 10 to the power of 3. So this will be 10 to the power of 5, and this will be 10 to the power of 7. Same as we did before, these plates need to label at the bottom your initial batch number, bacteria, and also the date. Because we're going to do a dilution. So what's the date today? 25th. But very important is your dilution factor. Okay? And always remember the final dilution factor of the plates is 10 times more than the tube dilution because we are adding 100 microliter. We are not adding 1 ml. If we add 1 ml, that will be the same. But why we cannot add 1 ml? Because it's too many liquids, or too much liquids on the agar plates. Okay? Now, there is something we want to say also. Today we are using the tips. The pipetting tips. So every time when you do a serial dilution, between tubes, tips has to be refreshed, which means you need to use new tips between the tubes. Okay? That means new tip for each dilution. So let's say all these tubes are ready. Now we're going to add onto these plates. When we add, always add from high to lowest dilution factor. Only need 1 tip. Why? Because when you first tip, you add it here, it's diluted 10 to the power of 7. And then you use the same tip to do the second one. It's not going to affect the results too much. Think about it, you already diluted 10 to the 7. And then you do the next one is 10 to the power of 5. It's not going to carry on too much. Then from 10 to the power of 5 to 10 to the power of 3rd, it's not going to carry too much too. So, only 1 tip to add these each. Okay, usually a drop. But you cannot do the opposite direction. If you're doing the opposite direction, you need a new tip every single time. So that's why we do high to lowest dilution is only 1 tip. That saves lots of the material. Now we're going to using is a spreader. The spreader looks like that, L shape. The spreader, how are we going to do? Same thing. Spreader from high to lowest dilution. Only 1 spreader. Okay? From here you spread it, go here, and then go here. Then you need to be let them be dry a little bit, stack it together. I think today we're gonna have about a week, so put these on your personal rack. Ideally, we can incubate 35 degrees 48 hours, but next week will be very messy. So, with this temperature around like 25 degrees Celsius with E. coli, you just put on your personal rack. We wait for 4 days. We'll count next Tuesday. Okay, that will be the plan. So, leave 3 plates in your tray. We will say for how many days? 5 days. This is approximately 22 to 25 degrees Celsius. Okay, so we're gonna do the counting. So that's what we have for today's work. Now, before I show you how to do, let's go over the results, what we have. First thing first, you have your nutrient broth, everything should grow. No question. Second, you have your blood agar, everything should grow. Because nutrient broth agar is a support media, blood agar is a enriched media. They should always grow. Then you can take out your blood agar like this to see the hemolytic. I don't see anything special from my sample. So it should be gamma hemolytic. Sometimes E. coli maybe show a light beta hemolytic. Okay, now look at your mannitol salt agar. The ideally results mannitol salt agar, E. coli should not grow. Why? Or E. coli should be light grow. Because they're not resistant to the salt. And what you should see, some of your colony, like Citrobacter, should be seeing yellow colony. Why? Because of acid fermentation. And the mannitol fermentation showing you yellow color. That's what it should be looks like. Macanthi agar, ideally what should be? E. coli should grow. Bacillus subtilis and the Citrobacter should not grow because those are gram-positive bacteria. But I don't know why it will grow. It should not grow. And your Macanthi agar should have only E. coli grow there. Bacillus subtilis absolutely is not grow because that's gram-positive bacteria. Okay? So, the results is not very good because it's been a while. Now, during the exam 1 review, which means midterm exam lab review, I'll tell you the scenario, what it looks like. I'll give you example of Staphylococcus aureus and E. coli, then you go ahead to fill the table. And during the review, we're going to re-emphasize it. Is that okay? Mm-hmm. Today, you can take the picture, just to record whatever you got on your notebook. If you want to do the bonus points, like I said. Because this is being a little bit older and it's hard to see how we're going to do. Okay, next one. Same as before. I will be showing you how to do spread plating. Again, before you do, wash your hands. Have your gloves ready. Have your dilution tubes, plates, a bacterial culture, spreader, and also some paper towel you need to be ready. And then let's all come here and I'll show you how to do it. So we're going to do a spread plating. This is basically a new thing to you guys. And we have lots of material. Generally speaking, compared to what we did before. So let's do it step by step. First of all, these plates are just coming from the refrigerator. It's very wet. So what you should do is all the bottom needs to be dry. Otherwise, you cannot label because the Sharpie marker will be faded. Second, I don't want to cause condensation on the agar plates, will be affect the results. So, the inside of the agar plates, you also should dry it. Because otherwise, those liquid may be cause condensation, which means your colony could be damaged. Okay, so inside, you also got to do a very gentle clean. Not the agar, I mean the lid. Okay, so inside also clean very well. Then what we do? We'll do a label. Label the plates first. On the side, your name, initial, bench number. Let's say E. coli. This is we're gonna do today and date 0925. That is remind you how many days we did incubation. Don't forget the important thing is the final dilution factor of the plates. Today we do 10 to the power of 3. And second one, we're going to do the same. See, it's already wet, still wet. So 10 to the power of 5. And then the last one, same thing, but dilution factor is 10 to the power of 7. Then stack like this. And then you can flip it over. It's the same thing for the tubes. The first time you do, I don't know whether you're going to be labeled. You can label your tubes if you're not familiar with the dilution factor. Okay? Now the other things. We want to make sure this pipette is at 100. Sorry. Make sure it's at 100. Sometimes people don't see it. Maybe 50 you start to do, then it's not going to be accurate. And also the tips, make sure this is a 200 microliter tips, not those big ones. Those big ones is for 1 ml tips. Okay? These are things already. Then what we do? We still need the fire because we want to make sure it's an aseptic environment surrounded you. So we still need to turn on the fire. This way. New one. Can I have a new one, thank you. Well, these are not good. Do we have a fire today? Maybe not. Maybe no gas. Sometimes no gas.
No gas.
No gas? Okay. Then we don't need it. That's okay. Okay, if we don't have gas, what we do? We still can do. First of all, you're going to arrange 10 to the power of 7, 5, and 6, or 5 and 3. Okay? If we don't have gas, we still can do it. But you have to do a little quick. Tips, you know how to do it. Is that right? This, shake 15 seconds. Let's say you shake it. Let's go from this direction, okay? So, this have to switch the tips. So put it here. Shake 15 seconds. Putting a second row. So that means it's finished. Then you take it out. Switch it. Shake 15 seconds. Then the last one. And shake 50 seconds. That's ready. So how we do? From here, gonna go here. So place it right here. We're gonna add it first of all. Only 1 tips. Because we are from high to lower dilution. It's only 1 tips. We shake it, let's say. So here. If you finished, put it at the back. Second. Last one. Then, ready to do the spreader. How we do the spreader? This is a disposable, sterilized plastic spreader. So make sure you only open a little bit, okay? Take 1, only need 1. From high to lowest dilution. How we do it? Have your hands here. This is called the spreader plating. Averagely spread on the surface. We've been this a lot in the lab, maybe 20 years. Second one. This is what I said. You can use one spread because when you carry on from 10 to the power of 7 dilution, it's very limited amount. Okay, the last one goes here. Dry a little bit, flip it over. Because the condensation, don't want the moisture to damage it. This, put on the tray. We took out next week, Tuesday, you should have a very good colony in some of the dilution factors. So this is what we're gonna do today. Okay, and all these used dilution tubes is the trash. So give to Rion. Very important. This guy return to the front desk and don't contaminate it. Otherwise, the E. coli is no longer E. coli by the end of the day. It's mixed with everything. Then you will see maybe yellow, red colony in some of the plates. Okay, this EC return to the front desk. Your dilution tube is biosafety, biohazard trash. Okay, so give to Rion. We're gonna put on the side and we'll autoclave it. Okay, that's all we have.